Well, hello and welcome to everyone joining us for this X-Talks webinar. Today's talk is entitled Going Beyond ATS ERS 2019 Guidelines for Your Respiratory Clinical Trials. My name is Corey Stanton and I'll be your X-Talks host for this event. Today's presentation runs for approximately 60 minutes and the webinar includes an interactive Q&A session with our speakers. To make the most of this session, feel free to submit questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to as many as possible. This chat box is located in the control panel on the right-hand side of your screen, and if you require any assistance, please don't hesitate to contact me by sending a message using this very same chat box. Please note that all participants are in listen-only mode and that this event will be recorded and made available for streaming on xtalks.com. At this point, I'd like to thank Clario, who developed the content for this presentation. Clario is a leading healthcare research and technology company that generates the highest quality clinical evidence for their pharmaceutical, biotech, and medical device partners. They offer comprehensive evidence generation solutions that combine eCOA, cardiac safety, medical imaging, precision motion, and respiratory endpoints. Clario's science, technology, and operational experts have helped deliver over 26,000 trials and contributed to over 800 regulatory approvals in more than 100 countries. For more than 50 years, Clario has provided deep scientific expertise and the broadest endpoint technologies to help transform lives around the world. And now I'd like to introduce our first speaker for today's event, Kevin McCarthy. Kevin has worked in the field of lung function testing for over 50 years, with nearly 40 years as the manager of the pulmonary function laboratories at the Cleveland Clinic Health System. Kevin's research experience includes managing the pulmonary function quality control in 2 NIH-funded registries. He has published extensively on pulmonary function testing and is a past member of the ATS PFT committee and served on the ATS ERS task forces to update the spirometry and lung volume standards. Kevin, I'm going to pass things over to you now to introduce our featured speakers for today's event.
Thank you, Corey. I appreciate that. With me today, I'm pleased to have Dr. Phil Lake. Phil is a respiratory specialist at Clario. He's held various positions in drug development over the last 25, 26 years, predominantly focused on respiratory drug development. He has led the respiratory strategy at a number of CROs and held the positions as head of the European project management for 2 of these companies. In addition to classical respiratory endpoints, his recent focus has been the use of lung function endpoints as a biomarker in rare diseases and in neurodegenerative disorders. And Phil has been pivotal in the first centralized overread studies and is committed to driving improvement in lung function testing through better understanding of data plausibility and variability to generate more reliable data. And with us today also, I'm pleased to have Benoit Guyvers. Benoit is a product manager of the clinical research portfolio at Artique. He has been leading the efforts on Artique's risk-based monitoring system from concept to solution. He has a background in human health engineering and a passion for impactful digital health solutions. So without waiting any longer, let's jump into it. The next slide here is basically not moving. Hang on one second. There we go. Thank you. So we have sort of a provocative title for this talk, and it's actually, it'll make sense by the end of the talk. There are limitations to relying solely on ATS, ERS, QC evaluation in clinical trials. That's been the foundation of putting a stamp of good quality on data or poor quality. And it wasn't until recently that we found that that is a pretty limiting view, that you need to expand your view and ask the question, does this make sense? Does it respect the laws of statistical and biological plausibility? And so we'll talk about that, and I'll give you an example of one type of error in spirometry in clinical trials that we've recently been doing a lot of work with. And then Phil will be talking about the impact of data plausibility review on clinical trials, and he'll show you some examples of that. And then finally, we'll wind up with Benoit talking about AI and data plausibility, and then we'll spend a little bit of time discussing how we can leverage AI and the data plausibility approach to reduce costs and accelerate timelines in respiratory clinical trials. So for those of you that are new to spirometry or new to clinical trials, I'll spend just a moment talking about the ATS ERS standards, because that's what we're talking about here. Unlike most diagnostic tests, pulmonary function laboratories and the measurements that are made there really have no accrediting body. And so the ATS, the American Thoracic Society, and the European Respiratory Society have sort of stepped up to the plate, so to speak, and periodically they write standards or update those standards on how different pulmonary function measurements should be made. And so in those standards, actually what they do is they review the literature periodically, and when enough published data suggests these standards could benefit from some tweaking, they'll convene a committee that will sort of review all that literature and then make some recommendations for change, which eventually get published as the standards. And they cover things like the devices that make the measurements, how accurate they should be, how they should interact with the person who's making the measurement. From the patient's perspective, they publish in that standards a list of conditions which you should consider relative contraindications. That is, you weigh the risks and benefits before you have the patient undergo that procedure. Now, the risks aren't great, but occasionally, you might want to know that your patient is in danger of increased danger of having a syncopal event, and you might, if you don't need the measurement, probably ought not to get it if that condition exists. So they spend a little bit of time, or actually a bit of time, talking about how the measurement should be done. And that's where I would encourage everybody who does spirometry to take a look at that section in the 2019 guidelines. So there's going to be some big changes you'll see, and we'll talk about those in a moment. I'll talk about how the data should be reported, and finally, give us some tips on how we can make sure that quality measurements are made day in and day out, what steps you can make in a quality assurance program. So this is a quote from the 2019 guidelines that I think highlights the situation that we're in clinical trials, that these standards that are in this document really represent the minimum criteria that must be met for clinical spirometry, but it might not be sufficient for all settings. And those two settings, research and occupational surveillance, that it points to really rely on repeated measures, looking for tiny changes in those repeated measures to answer questions about, in research, is this drug working, or is this drug safe for the lung? Or in occupational surveillance, how is this healthy lung reacting to this potentially toxic environment? So in the 2019 standards, and I promise not to spend a lot of time on this, I do want to give you some of the important changes. One of the most important was to take a look at the two most important parameters in the measurement, the FEV1, which is the forced expiratory volume in the first second of that forced exhalation. So it's what fraction of the total volume was exhaled, and the forced vital capacity, that is the total volume exhaled, but not just the total volume, it's total volume from full inflation. Both of these measurements should start from full inflation, otherwise you could be reporting values that are lower than what you'd like to be seeing. Now, one of the more important changes, and I might be biased on this, is the addition of a new phase to spirometry, and I'll show you that in just a little bit. But to describe it, if you were to picture this, if I have you take in a big deep breath and blast it out all the way, at the end of the forced exhalation, that's where the spirometry effort used to end. Now, the new standards say keep the patient on the mouthpiece and have them coach them to rapidly inspire back to full inflation and measure that volume. And we call that volume the forced inspiratory vital capacity. And that's important because now, for the very first time, we can compare that FIVC to the FVC that preceded it, and we have an objective means for evaluating a really important question. Did the forced exhalation start from full inflation? And that's what I think is so important, because we're finding out that routinely across the globe, there's a lot of people that they don't know it, but they don't coach to full inflation properly. And I'll tell you more about that in just a second. So if we look at the standards, and this is a very busy slide, but let me just summarize it briefly. There's 5 iterations, 4 before the most recent ones. Not one of those gave any guidance as to how the operator is supposed to coach to full inflation. But if you look at the 2019 guidelines, you'll see very specific instructions on how the operator should coach to full inflation, as well as the phase 4, that rapid inspiration upon completion of the forced exhalation. You'll see that. So why is that important? So on the screen right now, we have 2 volume time tracings. And the tracing on the left, you can see that it shows a rapid inspiration up to full inflation, or rapid inspiration, and then you see a very forced exhalation. And that where the red arrow is pointing is that transition from inspiration to forced exhalation. In my opinion, when I look at this, I see that there's unlikely that the operator really saw that the patient was at full inflation. More likely, what I've seen evolve is a way of giving spirometry instructions or coaching that goes like this: taking a deep breath and blow. And they'd use the wording and the cadence to be about the same with every effort. And what that does is it gives you good start of test. That is, you don't have any start of test errors because the patient is transitioning very rapidly from inspiration to forced exhalation. And if you deliver that the instructions with the same cadence, you get repeatability demonstrated across efforts within that measurement. Now, I'd like you to look now at on the right-hand side of the screen, and we see a volume time tracing that looks subtly different. Where you see the red arrow pointing, you'll see sort of a rounding of that, the transition point from inspiration to forced exhalation. And what should be happening right there is that the operator should be vigorously coaching the patient during that inspiration, and close to the red arrow, they should be going more, deeper. And when they see that the patient is giving apparent inspiratory effort, and they glance over the patient's shoulder at their testing screen and they see this rounding there, no further volume change, that's when the command to blow is given. Now, the danger is that if you take too long as an operator before you give the command to blow, inevitably patients will leak out a little bit of air. And if they leak out too much, that can invalidate the FEV1 from that effort. So I think that's why we got to where we're at, where we have a lot of people doing the technique on the left and getting what looked like, through the lens of the 2005 standards, really good data that is acceptable and repeatable. Now that we're doing this next phase of spirometry, we get this. This is what we're seeing. So here's 4 efforts from a measurement. It came out of an actual clinical trial. And these, you notice the volume time tracings showing regular tidal breathing, we call it, then a deep inspiration and the forced exhalation with a very sharp transition point like I just showed you. Now, this busy slide, let me summarize. 4 efforts, and we would say, if we looked at the repeatability of the FVCs, that wow, that data is fantastic. Had to have started from full inflation, right? There's only 110 ml difference between the smallest and the largest. Look at the FEV1.
Kevin, you need to move your slide on. We're stuck on the previous one.
Oh, thank you very much, Phil.
No trouble.
Boy, it got me again. There we go. No, I appreciate that. Yeah, I'm having a little difficulty with that. But so repeatable FEV1s, repeatable FVCs, and those are the 2 most important parameters. And repeatability, we used to think that meant this is really good quality data. But now if we look at the new number in spirometry, the FIVC, that's that volume that they inhale rapidly at the end there. That shows us that each one of these efforts started from about a half a liter below full inflation. And given the FEV1 to FVC ratio for this particular patient, that means the FEV1 was understated by around 0.4 liters, 400 ml. That's big. In a clinical trial, that could be sometimes double the treatment effect of a drug that's doing well. And so this is a really important problem. And this kind of summarizes what we hope to see, that is tidal breathing, deep inspiration, and notice how rapid it is, but notice how it starts to transition to a gentler slope. And that's where the rigid thoracic cage is really expanding, and it takes a little bit of work to do that. So that's the effort part of the inflation. That last 5% for patients is a little bit difficult. And then when the operator sees that sweet moment when they show no further volume change while they're still inspiring, then phase 2 goes into play when they give the command to blow. That proceeds until phase 3, where they demonstrate that they're empty. And then phase 4 is where you pick up back again with vigorous coaching back to full inflation. And if we look at this here, the things I want to draw everybody's attention to is The end of the forced exhalation is actually the start of the forced inspiration. So they share that common measurement point. If you look up at the top, that's the other common measurement point, full inflation. So those two volumes should agree with each other, and that's what the standards tell us. On the right-hand side of the screen, this is something I need to show you because it's important for the next couple of slides. This is a representation of what's called the flow volume curve. And it shows a very rapid rise to a sharp peak on the upper part of this graph above that hatched horizontal axis. And then after the peak, you see a reduction in flow that comes, it actually describes a kind of a concave shape. This tells us this patient has diseased airways. At the end of the forced exhalation, we see now below that dashed line, we see an upside down haystack appearance of the inspiratory flow volume curve, which looks so different than the expiratory because of the physiology of how air moves in and out of the lungs. Up above it, the driving force that is pushing air out of the lungs is also pushing to narrow airways. It actually exerts a closing force on the walls of the airways, and the airways on inspiration are actually being pulled open. So if we look at the next slide here, this kind of shows us the flow volume curves in two different scenarios. The one on the left is somebody with no airway disease. We know that because after the peak, the line drops down to the horizontal axis in a nearly linear fashion.
Kevin, we've lost your slide again. So you just need to.
Geez. Okay, very sorry.
Perfect.
I should have somebody else controlling these. Anyway. This on the left-hand side is the normal flow volume curve. On the right-hand side is that sharp peak. And now we see a concavity to that descending limb of the expiratory flow volume curve, telling us this patient has airway disease. And then we see that upside down haystack appearance of the inspiratory loop, tells us it was taken in with good effort, maximal force, and it ends to the left, unfortunately, of where that forced exhalation started. But that's actually the purpose of this. It's to alert the operator that on that effort, when you collected it, you really didn't take the patient to full inflation first. So it's something that's easily correctable at the time of testing. Unfortunately, that's not what our hopes about how it would be used. There we go. Have not borne much fruit. Some sites have used it as a learning opportunity to say, okay, my technique was not as good as I thought it was. I need to adjust that. And then they end up doing what we see on the left-hand side of the screen here. This is actually, both of these come from the same patient. On the left, it was from baseline. And this shows us the good, brisk inhalation that comes very close to the starting point of the forced exhalation above it. That tells us that even though they don't match exactly, it's close enough to say, yes, we would accept this data. It's good quality data. On the right-hand side of the screen, this is a measurement from the end of the treatment period that has us scratching our heads, because even though the expiratory and the inspiratory volumes match each other, notice how the shape of the inspiratory flow volume curve has changed. It's now, we don't see that upside down haystack. We see a very low flow. This is inhaled at about 0.5 liter per second, meaning it would take about 5 seconds for this individual to inhale all of that. And it's very easy to stop the individual at the end of where the volume matches where it started for the forced exhalation. And as a side note, patients do not want to inhale slowly at the end of a forced exhalation. On the contrary, they want to inhale as rapidly as they can. It only requires a little bit of coaching to get that last 5% back to full inflation in. So this is one example of a type of implausible data that can mess with a clinical trial's integrity, is when you have something that looks by ATS standards like it meets all the standards, when in fact, if you dig a little deeper, you're left answering the question, well, no, maybe it didn't meet that full inflation standard. This is something you'd want to correct. So now I'm going to turn this over to get a broader perspective of how implausibility can impact, excuse me, there we go, how implausibility can impact the clinical trial as a whole. And so Phil, I'll turn it over to you.
Thank you very much, Kevin. Hopefully you can see my screen now.
Yes.
Right, so let me take you through the next phase of this. So Right. So we've heard a little bit from Kevin about the ATS criteria and the drivers behind it. So what I really wanted to explain to you is why is it that if we just use ATS ERS acceptability criteria in isolation, that it doesn't actually guarantee really high quality data. Now, Kevin's illustrated that if you use ATS properly, you're actually more likely to get accurate data because you're actually doing the technique properly. But the thing is, accurate data doesn't just rely on ATS technique. And there's a few examples of it. So if you can imagine, for instance, if you test a patient, they've got perfect technique, everything's fine. Next visit, you can select the patient again. And instead of selecting patient 103456, you select 103457 in error because nobody refers to anyone as 103456. It's quite feasible that you start testing a separate patient. Now, again, there's no untoward intent there. It's just that you've selected the wrong patient. Now, at the end of that, you'll get ATS grading on that data. And in the database, you'll just have two values with perfect ATS criteria. So you're completely blinded to the fact that the data is potentially completely different and therefore, most likely inaccurate. Now, again, it's not just a switching patient that isn't picked up. So if a patient comes in, the ATS criteria has a whole load of criteria which require you to test a patient at trough drug level. So you wash out all the broncholators. They're not supposed to do exercise or smoke, eat, do other things, loose fitting clothing, et cetera, But you can imagine that the technique itself doesn't pick up any of that. It just picks up whether they're sticking to the inhalation, exhalation kind of progression. So again, you can see a sudden transition in the lung function based on the fact that they've taken their broncholator inappropriately, but the ATS criteria will just say it's acceptable. And this is a problem. And again, we come to another one, the kind of site manipulation of the technique. So Kevin, in his last few slides, was showing how some sites to trick the device into giving it a high quality grading, they're modifying their FIVC so it matches. So all of these instances are basically just areas where the ATS criteria will pass something as good, but in fact, it's not guaranteed. So you've still got variability there that is not potentially picked up. And then the other thing is about the consistent submaximal inhalation. So again, Kevin also showed that it's the cadence. So if you have the same cadence every time, where you do the inhalation up to a certain point, exhale, then inhale again up to a certain point, as long as that's consistent, the ATS criteria will say that it's acceptable. It doesn't mean to say that it's accurate. It just means it's consistent. So this is one of the drawbacks of the ATS technique if it's used in isolation. It doesn't pick up any of that. Now, there's a couple of other areas as well in terms of implausible changes to lung function or biological plausibility. So again, if you're just looking at the adherence to those strict ATS criteria, if you're actually testing a 60-year-old woman and she exhales to a plateau in 3 seconds, that's highly unlikely because that's a healthy young lung. So if that was an 18-year-old, you might accept it, not in a 60-year-old because of natural progression and aging of the lung. So none of this is really included in a basic ATS technique assessment. So I hope you can see that you need to go beyond that to really start looking at other elements if you really want to get accurate data. And so when we look at this, so if we start looking at the plausibility of the data, in addition to the ATS grading, what we see is between about 8 and 24% of data includes an element of implausibility. And so by that, I mean variability, which is not consistent with accurate data. And so on average, it's about 8 to 12% for typical studies, which is quite a lot of data when you consider it. When the magnitude of the errors may be small, but it's still quite a lot of data. So I wanted to show this to you graphically as well. So if we look at the chart on the right, so what we started doing at Clario was actually measuring the variability of patients' data to try and pick up some of those, to focus in on the patients who got, implausible or inaccurate data. And so we use this modified coefficient of variability it's variability of on-therapy lung function over time. And so again, with high levels of variability, so on this chart, each dot represents a patient, each vertical line represents a site. So we can see that we've got some sites with really low variability data. So below about 15% in asthma, we very rarely see any problem with the core technique. Once we get above that level, we see quite prevalent problems. So we can see in this one site here that the variability data is really quite large. The other sites in this kind of view are pretty good. But when we actually look at the ATS ERS compliance of this problem site, we can see the total number of unacceptable tests is only about 1%. And if we look at the rest of the study, we can see that the overall compliance there in terms of unacceptable data is still about 1%. So just using ATS alone will actually mask all the problems of this site until you actually unblind the study at the end, when you start to see the aberrant treatment responses, and then all your problems really start. So this is really the point. If we actually look at the plausibility of data as we go through, it gives us a second bite of the cherry, if you like. The ATS does the first bit of quality. The plausibility then kicks in to pick up the rest of it. So I want to give you a couple of examples of this. So don't worry too much about all of this. This is actually the same patient tested 2 weeks apart. Now, in the first test, we can see a classic COPD lung function pathophysiology. So 2 weeks later, we actually move. You can see the shape of this is completely different. So this has a whole series of pressure-dependent airways collapse because of the losing lung parenchyma and tissue in the lung. Here, we've got a profile which looks much more like a healthy, normal individual. Now, it's just not feasible to move from COPD to healthy. I mean, if you had a drug that do that, you'd be a billionaire. But this just isn't feasible. So what this represents is actually a switch in patient, between one visit and the next. Again, when we look at the actual values, none of this is actually flagged as being unacceptable. It's all completely acceptable according to the ATS technique, because both of these techniques are fine. Let's move on to another example. This refers really back to what Kevin was talking about in terms of the nature of inhalation before you blast the air out. Now, this is actually this is one patient measured 3 times. And so you can see their flow volume loops where they're breathing in and out. They take the deep breath in right the way up to the top. They blast out until they're empty. Now, in this case, they're not breathing back in again, as in terms of the ATS 2019. But when we look at the amount of inspiratory reserve volume, so the amount they breathe in on top of their Tyler breathing, which is what you and I are doing now, the bit, we can see in the first example, there's a reasonable level of this. In the baseline visit, there's virtually no inhalation. So they've gone from breathing normally straight into a. Now, if you do that, you're not going to be able to breathe much air out because you haven't breathed it in. And then the final endpoint of the study, you've taken a nice, really strong deep breath in. You've got a nice little plateau here, and then you extend into an exhalation. So we've got there what appears to be 1.5 liters of improvement between baseline and endpoint. The reality is that this baseline measurement has been reduced by at least 1 liter. And so historically, Kevin was saying earlier that if we have consistency between the 2 measurements, normally this means that the data is accurate. That was how people were trained. But we can see for this value here, the 1.1 liter is made up of 2 measurements half an hour apart. There's only 40 milliliters difference between them. And so this historically would be evidence that this started from full inflation and the data was good. All this really means, this consistency, is that your technique was consistent. So you had the same cadence as we were doing before. So it doesn't actually equate to accuracy. So let me show you another way in which we kind of start to plot data. So I introduced the coefficient of variability earlier. So here, this is the coefficient of variability is now on the x-axis, and we plot that against the change in FEV1 from baseline. So this is the change from baseline to endpoint for each patient. So each patient's a dot. So we know that once we get above about 15% variability, that the data becomes less reliable in terms of technique. So the patient I just showed you is this one here, this dot on the far right-hand side. 1.5 liters of treatment improvement, massive amounts of variability. And so you'll notice that these patients here, the magnitude of change is just completely implausible. So 1.5 liters, that could be 15 times a kind of an anti-inflammatory drug response. It could be 5 times a standard bronchodilator drug response. But these are the patients that are really driving the main problems to your study, which is why I like this chart. But if we look at the data longitudinally now, this is FEV1 and each visit, and each line is a patient. The patients that generate this data here with the massive variability, These are the kind of roller coaster patients that I like to call them. So again, they're increasing and decreasing by a litre and a half, 2 litres between visits. Now this is into the territory of growing a new lung and having it removed, growing a new lung, having it removed. It's completely implausible. You would not expect to see this kind of response to data. Now, when we actually review data retrospectively, so what we've done here is we've taken some data where we know there's problem patients in it. And what we've done then is actually we sort it according to the modified coefficient of variability. So this is the very worst or the very highest variable patients within the study. So it's about the worst 6%. So the vast majority is fine, but this is the worst data. But when we actually look at those patients and we look at the initial ATS ERS compliance by overread, we can see that all of them are acceptable or occasionally borderline acceptable. If we look at the change from baseline, these figures again are in this kind of fantasy realm of lung transplant kind of era. So we've got these massive changes in lung function. Occasionally, it doesn't always have to be increased, it sometimes decreases. But what we really know is that this data isn't reliable because we can't trust some of the data points in the study. So it could be the baseline, it could be the endpoint, it could be something in between. But again, when we actually start looking at this data as an individual patient perspective, and not just using it to ATS criteria, but looking, is there any evidence for submaximal inhalation? We then kind of have a very different view of the data in terms of its quality. So we're rejecting about 85% of that data. And it includes 2 patients where they've actually switched the patient by mistake in the middle of testing. So again, ATS doesn't pick up any of this. It's only really once you start looking in this nature that you start to pick up the problems. Now, you'll notice as well, there's 2 patients here which show acceptable after review. Now, again, this doesn't necessarily mean that the data is still accurate. What it means is that the data doesn't show any deviation from standard forced spirometry techniques. So this could still be related to either an abnormally good treatment response, or it could relate to the fact that the patient's taken a bronchodilator before they come in. So again, when you see these high treatment responses with high variability, good practice would be to actually check to see whether there is a mistake in terms of bronchodilator use that hasn't been recorded. So just to reassure yourself. So another way of testing data is we start looking at the FEV1 FVC ratio. Now this relates to the level of obstruction in the lung. So as your lung gets older, the level of obstruction increases in general. There's some conditions that will reverse that or prevent it. But if we look at the change in FEV1 and FVC ratio over time between visits, what we can see is that it's pretty unusual for that ratio to change by more than about 10%. So if we plot these plots here, which showing a greater than 10% change between visits, and we start to look at which sites are generating this, in this case, we can see this site A has the vast majority of all data that shows these completely implausible changes. Now, occasionally these things happen. You may get a patient switch, or there may be some extreme technique problems, but you'd normally not expect there more than 1 patient per site. So when we start to see this, it really starts to ring alarm bells. So when we looked at the data from that site A over here, we can see that the flow loop geometries all look remarkably similar. Now, the thing is, this isn't 1 patient. This is supposed to be 6 different patients. So again, this brings us to another element that we have in clinical trials. It's pretty rare, but we have kind of these mirror patients. So it's potentially the same patient has been tested multiple times. Now, sometimes that's permitted within the study, sometimes it's not. But what we started doing is actually looking for the similarity of patients, because again, all of these are ATS ERS compliant. So, when you're actually looking for variability, that's great, but similarity is a bit more tricky. So what we really do here is we plot a similarity index to show how similar the data is from the site. So again, the more patients you have in a site, the greater potential that you'll have a very similar patient. But so we plot a kind of best fit line, if you like, and we show then deviation away from that. So this particular site, you can see is a massive outlier compared to the trend of what we're expecting. Now, and Benoit is going to talk about this a little bit later, because this is actually based on numerical similarity. But with the AI approach, we've got the potential to add in flow loop geometry similarity as well, which really takes it to another level. Okay, so I've shown you all this. So what does it really mean, and what action do we need to take? So the reason I like this chart is it helps you to understand the impact of variability on drug trial outcomes. So this actually shows the benefit of a drug, either active or in favor of placebo. So the treatment effect, so this is a treatment difference in FEV1, and this is the number of patients we have in the study. So when you analyze, and the confidence interval of finding a treatment effect. So when we analyze all of the patients, we can see that the drug doesn't really do very much. And this is fine because the drug was discontinued. So we're not expecting it to do anything. But when we start to look at smaller and smaller treatment numbers, you can see that here, as the treatment numbers come down, these are different groups of patients, we can see that we get variable increases or declines. So, and in the small levels, we can have, a really good drug effect or a really bad effect in favor of placebo. Now, what this relates to is the distribution of some of those problem patients that we showed you earlier on. So you can imagine, just one of those patients which has, 10, 15 times the normal treatment response. If you have too many of those in one arm, you can start to modify the treatment response quite easily. And so this is the thing, in a big phase 3, technically, if you just want a mean treatment response, the powering of a study will often account for that variability. The thing is that if you want to see a subpopulation, so if you want to understand what the treatment effect is on those with high blood eosinophils, for instance, or if you want to find out how many patients actually decline by 10%, having a mean response isn't going to help you there. So literally, all of that data is going to be lost because of the variability. And again, if you've got a dose ranging trial where you've only got 90 patients per arm and you've got 4 different doses, you need to select the right dose. If you don't take into account this variability, the danger is you select completely the wrong dose and you move forward into the next phase with literally the wrong assumption. So you can waste hundreds of millions, if not billions, in this kind of approach. So let me take you to the final slide I've got. So we have this massive change in the way that we look at quality and a huge potential to then feed back to the sites about whether the data is good or bad. Now, our experience from the ATS ERS 2019 launch was that when we first started that, we fed back and were very strict on how we implemented it. But the amount of pushback from sites was massive. And a lot of them complained because the screen failure rates were going up. They found it really difficult to recruit patients. They were losing motivation. Now, we have a similar problem here. So we really need to think about this whole process as a change management concept. We need to think about how quickly we introduce this and how we take the testers with us so that they don't get overwhelmed by the pace of change. The other thing that I'm very cognizant of being in development for so many years is the law of unintended consequences. So you often do something with the best intent, but it has an effect which you haven't considered. So I'd like you to think, this kind of hypothetical example is that if we actually change the patients we include, so to only those that can generate really good data, we change the population that we're testing. And so, I can imagine an example where if you're trying to actually show an exacerbation trial or show improved control of the disease, if you only select patients who are good enough to do perfect spirometry, they're going to be well coordinated. And a lot of the patients that struggle with control are those that can't actually coordinate their rescue or the rescue medications or the normal medications, so they don't get drug into the lung, so they're underdosed. A lot of the new drugs that we're doing are injectable or oral. Now, they really help that kind of patient, and so they're likely to show a massive improvement. If you're excluding that patient population on the pretense of trying to get better lung function, you're potentially undermining the trial. So I think we need to it's just a word of caution, really. I think we need to have a discussion when you're thinking about running trials as to what is the best way to actually pace this so we actually don't slow recruitment down. We get all the benefits of what we're trying to do without any potential negatives. So I'll leave it there. I'm going to hand you over to Benoit, and hopefully he can continue this interesting topic and show you what's coming in terms of AI.
Yes, okay. So yeah, my name is Benoit, and I'll be talking the next 10 to 15 minutes about how AI can actually impact respiratory clinical trials and create impact there. I think AI has been hyped a lot in the last 3 to 4 years, especially due to the ChatGPT and the large language model technology. But as we all know, AI has been around for much longer in different forms as well. And there are already many examples of successful implementations of AI in clinical research with a proven track record. So that's what I'll be focusing on, with a deep focus on quality, and data plausibility. Before we go into some examples, again, I'd like to celebrate the acquisition of Arctic by Clario, which was recently announced. 5 years ago, Arctic started as a spin-off company from the University of Leuven. And in those 5 years, and even before that, Arctic successfully implemented and developed AI technologies in respiratory clinical trials. And with this acquisition, I think I see this as standing on the shoulders of giants. With Arctic's past, current, and future AI developments, I think all of those will benefit from Clario's expertise in clinical trials. For example, with Clario's scientific knowledge, we might validate new AI concepts faster, but also the Clario team can guide the Arctic team to the bottlenecks of clinical trials. So we can focus on what matters most. I think we can consider AI as a disruptive innovation. So why don't we look back at the past and see, okay, in the past, how were successful disruptive innovations handled and what drives successful disruptive innovation in the past? So I'm taking you back to the 1890s. Their innovators, they managed to automate elevators. And suddenly elevators were fully automatic. So the traditional elevator operators, they were not needed anymore. And people could just take elevators by themselves. However, being an elevator operator, that was still a job for a long time after. So why was that? Actually, it turns out people felt uncomfortable with the innovation and they still prefer to be guided by a human expert. And also other examples show us that disruptive innovation has a higher likelihood of succeeding when there are human experts there to guide us. And in the same way, we believe that Clario's expertise is the best way to guide the clinical research industry through these times of AI innovation. So I think that's a nice bridge or an anecdote from the past. Okay, so where is AI in respiratory data quality? So I'd like to start from the FDA and GCP guidelines on risk-based monitoring. I think regulations can be also a source of inspiration for innovation, so we start from there. And for risk-based monitoring, there are two aspects that we should take into account according to the guidelines. On one side, it's the participant safety, rights, and welfare. And on the other side, we have the data integrity. And for both of these aspects, we should do a continuous risk assessment to find out what are the risks. And then this should be the inspiration for a risk-based monitoring plan, which will help us to monitor these risks and also mitigate them in an efficient way. So let's look at spirometry data specifically. Spirometry, which are the two focus risk factors that we should take into account. First of all, spirometry, as Kevin and Phil talked about, it's an effort-dependent test. You really need to do your best to make sure you have the correct technique and also the correct outcomes afterwards. And also, not all study sites, they have the same expertise in spirometry. That's on the left side. On the right side, spirometry biomarkers, they're often used as an inclusion or an exclusion criteria. So it is a crucial data point, which also means that it's a potential point of manipulation. There might be data manipulation to meet the inclusion or exclusion criteria that could happen. So I think taking both these two risk factors into account, that's the basis of the risk-based monitoring approach that you can take. But okay, those are the risks. How do we mitigate these risks? For the first point, I think we should use the ATS ERS guidelines, as Phil and Kevin talked about, specifically for the quality control of spirometry, making sure the playing field is leveled by overreading the data centrally, so all data on a central location. There is a consistent quality control mechanism in place across all study sites. So there, the visual inspection of the curve data, that can be done by humans, but it can also be done by AI. There is also the option to have a combination of both, where humans are the elevator operators to guide us through the innovation of having AI there as well. The solution here is called ArcticQC, and in the next slides, I will be talking about ArcticQC and afterwards about the other solution. So what about the second point, the inclusion and exclusion criteria? I think there is a need for data plausibility check. Phil has been talking about it. We need to find a way to efficiently flag implausible data, but also suspicious patterns in the data. So it's about analyzing large data sets and spotting patterns. So that's a great use case for AI and smart algorithms. And then I think it's also a great use case for human experts to then focus on the flagged study sites and support the active follow-up, decide, is this indeed suspicious, yes or no, or do we need to take other action points? Maybe something went wrong indeed in selecting the patients. So there, AI can really guide human experts, and vice versa, human experts can guide the AI as well. So this solution is called Arctic RBM, and that will be the second example that I will be discussing. But we start with Arctic QC. So Arctic QC, it's the AI solution for overreading spirometry. And I'd like to link this to an example of AI technology that we actually all know. In fact, we've been training this AI technology without realizing it. And you can see it here. So when we want to log in into a website, we're asked to prove that we are human. And we do this by labeling data. And it's not a coincidence that it's also always road related or traffic related. We need to label, for example, traffic lights or a bus or a crosswalk. But what is happening in the backend is all those human labels are combined together. And then that large data set is used to train AI models to recognize these features as well on images. So AI models can also recognize traffic lights, for example. And that technology is particularly interesting for self-driving cars. So we've actually all been training this AI in the past. And the same logic can be applied on the concept of spirometry curve data. So what we can do is we can label artifacts that are related to quality issues. And then we teach AI technologies to recognize the same artifacts automatically. On this slide, we can see a couple of examples. So the green ones are examples of good quality spirometry. And in the red examples, we can see quality-related artifacts. So the AI model detected a submaximal blast or an obstructed mouthpiece or a hesitation. And this is then flagged and labeled automatically, just like a human overreader would do, but then instantly. So the decision or the output is made within less than 1 second, let's say. So how has this been developed and validated? So there was a large data set behind this with a representation of multiple diseases, and it was also labeled by multiple respiratory experts. In the years following its initial development, it has been validated in different contexts, and I would like to focus on one example specifically, which is highlighted here. In a clinical trial, the ATS ERS 2019 guidelines were used. So those are the latest guidelines, as Kevin explained. And both humans and AI provided an overreading label for the data. Afterwards, they were compared, and the great results were that in 91 and 95% of the cases, human overreaders and AI overreaders, they agreed on the labels for FEC and FEV1 respectively. So those results were already quite good, but then the question is, what about the other 5% and 9% where there was a disagreement? So we decided to invite top experts to blindly judge the quality of these difficult curves of disagreement. And we saw that these top experts agreed more with AI overreading labels than with human labels. So we are comfortable to say that Arctic QC is at least as accurate as human experts or human overreaders in judging the quality of spirometry curves based on the ATS ERS guidelines. Okay, what are then the benefits of having this in a clinical trial? The biggest benefit, I think, also talking to pharma companies, is the fact that it's instant. So imagine there's a patient in front of you and you're screening that patient for a clinical trial. The patient goes home after the test, and then it might take a day or 2 days for the overreading results to come back with humans. So there is a delay. And then it could happen that the quality is not good enough and you have to reschedule the patient. The patient needs to come back. Also, it's not only inefficient, but also it creates an additional patient burden. With AI, the results are instant, so you can instantly get the feedback on the quality and instantly redo the test if needed, if the quality is not good enough. The second point is that the results are consistent, so AI will not change its opinion, whereas humans might do that, depending if they had their morning coffee or not, before or after the coffee. So AI is always consistent, and that's a very important feature. Humans are not always consistent. Also, there can be a disagreement between humans. And finally, as I mentioned before, it is a validated technology to be at least as accurate as humans, which is an important factor to take into account, especially in clinical trials. So that was a part about Arctic QC. The second part is Arctic RBM, so stands for risk-based monitoring. And we actually take some inspiration from different industries. So let's look at the financial industry, for example. They have regulatory obligations as well. For example, anti-money laundering. They are obliged to check for any anti-money laundering. And how do they do that? They have this huge amount of transactional data, and they use AI to flag suspicious transfers. And these suspicious transfers can then, in the end, be checked by human experts to decide this is indeed suspicious, we should follow up on it, or there's something other going on, we can ignore it for now. So that's a bit what the financial industry is doing. We can actually do the same thing in respiratory clinical research industry. So we have a huge amount of spirometry data, and we can use AI to flag potential study sites that something might be going wrong on different levels. And then specific cases can be checked by humans to decide maybe this site needs a retraining, or this site needs a site visit, or we need to review additional data sources to rule out if there might be some data plausibility issues in the data as well. So the concept comes from, we can learn from different industries and apply the same learnings in clinical research. An example that Phil talked about before is the mirror patients. So in theory, one human should be represented as one study participant in a study, but it can happen that one human is represented in multiple study participants. It can either be a mistake or there can be more going on. But what can we do about it? So we can look at the FEV1 values, the FVC values, or we can do more. We can look at the morphology of the flow volume loop, for example, and consider this as some kind of spirometry fingerprint. And if there is a high similarity in of the same fingerprint in one study site, that's a flag that we can raise and say, hey, there might be a couple of study participants that are suspiciously similar based on the morphology of the flow volume loop. We need to check other data sources to rule out if something is going on here. So that's the concept of spirometry fingerprinting. We can actually use the same logic behind this algorithm and apply it longitudinally. So a participant at screening, it should look the same as the participant at randomization. In theory, you would expect this. There might be some small deviations or variations, but not a completely different fingerprint. If this happens, again, this can be spotted in the morphology of the flow volume loop, and AI algorithms are pretty good in doing this. This can then be flagged and checked by human experts to decide we need follow-up actions or not. Okay, then I think here I will be handing over back to Kevin for the conclusion.
Thank you, Benoit. I appreciate that. So I think that to summarize today's talk, I think we've presented some interesting work that kind of shows us that although the standard way of looking at the quality of spirometry measurements in clinical trials, that is looking at ATS acceptability and repeatability, is really good, but it doesn't catch everything. That you can have up to 8 to up to over 20% of the data that looks fine when you do that assessment, but really can be problematic and can really jeopardize the integrity of your study. And so when we look at these, we've developed techniques for looking at these, and when we look at measurements that are flagged for these for data plausibility issues, we often see that we can find the cause of that. And it's identifying the cause is actually really good, because particularly if you think about taking this concept and applying it early. Early on in a study, you can pick up problems and kind of nip it in the bud, so to speak. And I think that can go a long way towards saving time and money in a clinical trial. Embedding data plausibility within AI functionality, I think, brings it even closer. That is, it really speeds up the detection. It speeds up the process where you say, okay, there are problems, but we're not going to let it happen in our study. We're going to flag it and correct it as quickly as possible. And all of that leads up to the concept of really changing the way we've been doing clinical trials from the standpoint of like saying, yeah, we need tons and tons of patients and we need long study periods. In some cases you do, of course, but a lot of this can change. If you can really eliminate a lot of what historically has been present in clinical trials, the noise that obscures the signal becomes clearer, and it's easier to answer the questions that the trial is designed to do. So that's why we encourage you to contact us at Clario. We can help you de-risk your study, so to speak, using some of the insights that we've gained with our experience and the monitoring tools that we've developed. And we can empower you with insights that can help you evaluate these data inconsistencies or these anomalous data instances and figure out what's causing them as quickly as possible so that you can change that. And we want you to actually get into conversations with us to see if you can, if you have an interest in using this new exciting AI capability that we're going to be coupling with our overread and our plausibility to really, I think, become sort of the industry leader in running healthy respiratory clinical trials. And all of that, I think that the benefit of this, the sort of the pot of gold at the end of the rainbow is that we're entering into an era, I think, where we can see faster clinical trials, cheaper clinical trials, and help you make good decisions about how your trials should be run. So please reach out to us, and we'd be happy to have a discussion with you. So I'm going to turn it back to Corey now. I think we've reached the Q&A section of section of our talk today.
Yes, that's right. Thank you very much, Kevin, and thank you, Benoit and Phil, for your presentations. Very insightful information from you all. We do have time for just a few questions as part of our Q&A session here. We have received from the audience already, but to any audience members who have questions, feel free to continue to submit them using the questions chat box, because the team at Clario will have a chance to follow up with you after the webinar. But I did want to get to the questions that we already received throughout the presentation. This first one, I think, is probably best suited for Benoit to answer, actually. This audience member is asking, what happens if a study site actually disagrees with an AI-based overreading? You know, can the study sites discuss any specific cases with Clario to resolve discrepancies?
Yeah. So it's basically the project team. They still have the expertise on respiratory level, but also on AI product level. And then the workflow is essentially the same as with human overreading. So we'll try to understand the site's perspective and also explain the AI model's decision-making process and eventually come to a resolution. So it's pretty similar to what we're used to.
Okay, great, excellent, thank you. This question here, probably best suited for Phil. This audience member is asking, what does Clario do in a situation when you identify any anomalous data?
Yeah, I mean, that's a good question. So I mean, the first step really is to discuss it with the sponsor. So I mean, you know, ultimately it's the sponsor that's responsible for the site quality. So if we pick up issues, again, it can be a whole range of things. So we really need to discuss with them what we think the best steps are to progress from that. So as part of the CDS program, I mean, we actually kind of take on board either email communication with the site to help them, you know, explain to them what they're doing wrong, or we have one of our training group contact them and walk them through the issue and exactly how they can fix it. So, you know, the idea is to identify it early, step in early and fix it, you know, so that, you know, you don't generate any more damage to the trial as you go forwards.
Okay, great, excellent, thank you, Phil. I'm just checking on time here. I think we probably have time just for one more question here from the audience, and actually perfect timing. I think, Kevin, this one is probably best suited for you. This audience member is mentioning that they had a prior asthma trial which then had generated some unexpected results, and they're wondering if it's possible for Clario to review data retrospectively to provide insights, or what exactly is the sort of timeline looking like when you review data?
It's actually both. I mean, retrospective is certainly possible. The tools that we have developed can be used over large data sets to sort of flag anomalies, and we can go in and then use the human part of it to evaluate what exactly is going on. As Phil had indicated, I think the strength of this as a tool really lies in the almost as close to real time as you possibly can get, identifying these things early on. But of course, that retrospective review can actually help you learn lessons about clinical trials. It kind of like points out some things that were maybe could have been done better or could be done better in future trials. So.
Okay, great, excellent, thank you. We did have some more questions coming in from the audience, but unfortunately we have reached the end of our time for the webinar today. So for our audience members who have submitted questions that we couldn't get to, or if you have any further questions, you can get in touch with the team at Clario using the contact info currently up on the screen, the email address there, science@clario.com. Also, if you're watching this recording and you have any further questions, please use that email to get in touch with our team, and they will have a chance to follow up with you after the webinar. I do thank everyone once again for participating today. I'm adding a link in the chat box right now where you'll be able to access the recording of today's events, usually available in about 1 to 2 business days, and you can share this link with your colleagues so that they may register to view it as well. For those of you who are interested in hearing more from Clario, you can use the second link I've added to the chat box to register for their next webinar. It's titled A Holistic Approach to Parkinson's Disease Endpoint Data Collection, Efficacy, Safety, and Quality of Life, and that's coming up next month, Thursday, May 16th. You will also be receiving a follow-up email from XTalks that will provide access to today's recording. And as we wrap things up here today, please note that a survey window will appear on your screen. Your participation in this survey is always appreciated as it helps us to improve our webinars. Now, please join me once again in thanking our speakers once more for their excellent presentation today. Kevin, Phil, Benoit, really appreciate your time, really appreciate your insights. We hope our audience found it informative, and we wish you all a great rest of your day. Take care, everyone.
Thank you.
Thanks, everyone. Have a good day.
Thank you. Bye-bye.